Good afternoon. Turned out to be actually kind of a nice day today. It's kind of a nice surprise. Only announcement I have is that don't be afraid to stop me or raise your hand if I say something goofy in class or if I say something wrong. A student pointed out, thanks to that student, that I called ciprofloxacin Keflex, which it was the cephalosporin trade name. Keflex, one of the cephalosporins. Ciprofloxacin usually we just call Cipro. That's a trade name. So that was a fluoroquinol example of a fluoroquinoline. I made that note when I posted the video below the video, but for those of you in class, just I goofed up there, said the wrong thing. Once I get like an hour into lecture, I start, I'm sure you get tired of listening to me. I get tired of talking as well. All right, so we went through a kind of long list of antibiotics last time. Again, pay particular attention to their mechanism of action, any big advantages or disadvantages to their use. So we talked about some strange side effects, to a lesser extent, how they're administered or the kind of organisms they target, how narrow or broad their spectrum is. But again, pay particular attention to their mechanism of action. So we're kind of ended last time thinking about antibiotic resistance. And we said that often those antibiotic resistance is, we think, most often passed by some form of horizontal gene transfer, most often conjugation. We think that this is pretty rare, but we think that it does happen. So where we left off, we wanted to talk about specifically how does an organism, what does it actually do to enable it to live in the presence of antibiotics? One of my colleagues that I used to work with, he used to say, teaching students about resistance is futile. And what he meant by that was it gets kind of confusing because, again, you have the host, you have the bacteria that's in the host, and then you have the antibiotics. So it's part of just keeping track of what's doing what, right? So if we think about how bacteria acquire the ability to live in the presence of antibiotics, we're going to talk about 3, depending how you count it, 4 different mechanisms. Your book talks about these mechanisms as well. It counts it a little bit differently. Your book lists like 7 different mechanisms. So it's mostly the same thing. I'm just kind of counting it a little bit differently. All right, so one way is bacteria, if they can limit the access of the antibiotic into the bacterial cell. So that just kind of makes sense. If the bacteria can keep it out or pump it out once it comes in, then it's less likely going to be affected by the antibiotic. All right, so one way that they can do that is by altering their porin structure. So if you remember, go back to your structure of the gram-negative cell wall. In that outer membrane, they have these protein channels called porins. And we think that if the cell, if it can acquire, some kind of point mutation in that porin structure, then it can be a little bit more particular about what it lets into the cell. All right, so again, the less drug allowed into the cell, the better chance the cell is going to survive exposure. So it just becomes a little bit more impermeable. Okay, that's one possibility. Another possibility is once that drug comes in, And I think when I unmute this, is usually when I get feedback, but maybe not. Okay, wait. Oh, all good now. All right, we'll try it. Okay, good. Okay, so we think that a lot of cells have ways to get rid of toxins that they're in the presence of. So they just kind of modify these pumps to be able to spit back any antibiotic out that they are in the presence of. So we think that this is one mechanism that a cell can become resistant to a wide variety of antibiotics. Tuberculosis, Mycobacterium tuberculosis, there are strains of this that are basically resistant to any antibiotic that we've thrown at it. And this may be a mechanism that can happen. All right, so the second mechanism, we have talked about an example of this already. When the bacteria can produce some kind of enzyme to inactivate the drug. All right, so the example that we've already talked about is some bacteria acquire the ability to make beta-lactamase. Remember what that enzyme does? It actually, here's that beta-lactam ring. It actually cleaves that ring open right here. It's a decarboxylation actually, but when that structure opens up a little bit, then it won't fit into the active site of the transpeptidases that's reforming those amino acid bridges in the peptidoglycan structure. All right, and there's lots of different kinds of beta-lactamases, but again, we think the information to make that enzyme is often passed on a resistance plasmid by conjugation. And I tried to search through the literature a little bit to find another example of an enzyme that could be produced. Question? Oh, the screen is frozen. Oh man, if it's not one thing, it's another today. Thanks for pointing that out. Oh man. Let's see. Let's try to share the screen again here. Uh-oh. All right. Sorry, people at home. Let's try this again. Oh. Lovely feedback that I get. Okay. All right, we'll try it again. May have to do this recording all over again. All right, thanks for letting me know. All right, so what I was saying is that I spent some time looking through the literature trying to find another example of a an enzyme that would modify, sorry, let me just check and see if I'm recording this or not. Oh, okay, all right. Another example of an enzyme that might modify an antibiotic. And there have been some that's been discovered, especially within targeting antibiotics that seem to target the ribosome structure. There are other enzymes that would, for example, change a functional group of the drug. So for example, here's just an example I found, gentamicin. Remember, that's an aminoglycoside. This was just a paper out of Nature, I think, that I found, but a group had found some enzymes that just modified some of the functional groups, like the amino group or a hydroxyl group here, sometimes just by adding a phosphate to it or by adding an adenyl group to one of those groups. Just kind of a slight modification of the drug restricted its ability to bind correctly to the ribosome. So it was making, the bacteria was making some kind of enzyme that made the drug unable to bind to its target spot. All right, third mechanism is not modifying the drug itself, but if the bacteria could modify part of its own cellular structure so that the drug can't bind to it. So some examples here, ribosomal RNA methylation. So remember, ribosomal RNA is what makes up a ribosome. And if the bacteria can make some kind of enzyme, like a methyltransferase, that will add a methyl group to parts of that ribosomal RNA, then that's going to keep the drug from binding. Okay, so keeps the drug from interacting with the ribosome. Okay, and that doesn't seem to affect the ability of the ribosome to make proteins. It's just kind of a small change here or there. We've also seen instances where bacteria have been able to, or they've had a point mutation in the actual binding site of the drug to a particular enzyme. So you had a point mutation in the enzyme, like a point mutation maybe in DNA gyrase, that wouldn't affect the ability of it to, relax those supercoils in replication, but the ability of the quinolone, for example, to bind to the DNA gyrase would be impacted. Or like rifampin, point mutation in RNA polymerase, rifampin can't bind to it. It still can transcribe the DNA, but there's just some small change somewhere that would alter the binding. In the fourth mechanism, maybe you wouldn't really call it a mechanism because it wouldn't be something that would be heritable to the next generation of bacteria. But we do know that microbes, at least some kinds of microbes, tend to be able to switch into kind of a dormant non-dividing state. And it's not clear to us how they're able to do that. But especially say when they're forming biofilms, big mats of bacteria tend to act a little different. They're not really actively growing. They're just sort of persisting. Okay. Oh yeah, I wrote that right here. Persister cells and biofilms. And we know when cells aren't actively growing, a lot of antibiotics don't work very well. For instance, all the beta-lactams need an actively growing cell because it's when their cell wall is expanding that they act to weaken that cell wall. So the question is, well, what do you do about that? Is there anything that we can do to make bacteria kind of come out of this dormant state? I happened on this article a few years ago, 2017. It was this article of some research that some scientists at MIT were doing. And they were trying to find ways to kind of jumpstart bacteria that weren't actively growing so that antibiotics would be more effective. And it made me think of, well, what do you do for a kid that's sick, trying to get him to eat, right? Sometimes it's a hard thing. You don't give him spinach. You say, hey, how about a popsicle to try to get him to eat? Well, they kind of did the same thing to try to get bacteria to jumpstart their metabolism. Not only did they give them a sugar source that was readily available, but they found they also had to give them some kind of available electron acceptor. And oxygen didn't seem to do the job. They give it some organic oxygen electron acceptor to try to jumpstart the metabolism and get them out of this kind of dormant persister state. And they had some success with that. So I thought that was kind of interesting. Look at that if you're interested. All right, so those are just some ways that we know specifically that bacteria can survive presence of antibiotics. I happened upon this little graphic and I thought that it might be helpful to try to see if you can identify. So I listed the different mechanisms down here. Let's take a minute or two and see if you can match up what the graphic is describing, like which one of those mechanisms it's describing. So just take a second and then we'll go through the answers together. All right, let's see how you're doing here. So this one here, efflux pump. So it's pumping out the antibiotic there, that's number 3. Decreasing the membrane permeability, that just means that it's not allowing the antibiotic in, that's number 1. Modifying the binding sites of bacterial enzymes. Okay, so that's number 5 here. Where previously the antibiotic bound to the site and now it's not able to bind. That's what it's trying to show. Altering the porin structure, that's number 2. So here's the porin channel, altering it so that the antibiotic's not able to come in the cell, and modifying the drug itself. Here's the drug and now it's changed a little bit. So just sort of a nice little exercise there. Okay, so antibiotic resistance, it's a problem. Scientists worry about entering a post-antibiotic age. So are we going to get to some time where none of our antibiotics are working anymore? That's kind of a scary thought. We need to try to be working either continually finding new antibiotics or finding other ways to fight bacterial infections. If you've ever read, anybody read Sinclair Lewis, this book, Aerosmith? Okay, put that on your summer reading list. It's a classic. It's a story of a doctor, like early 1900s, how he was, how he became a doctor. And then once he became a doctor, he set upon some research to try to use bacteriophage to kill bubonic plague. Of course, this was in the pre-antibiotic age, and it's really an interesting story. This was before that was possible. We may get to the, this is something that we should continue to do research on, maybe using viruses to kill our bacterial infections. Maybe we need to learn how to manipulate our microbiome to be better at keeping out intruders. Hopefully we can find other ways so we're not so reliant on antibiotics. All right, any questions about anything? Chapter 10, antibiotics. All right, let's move on then to chapter 14. All right, so we've spent a little time trying to think about how to kill microorganisms. We're going to look now at kind of the process of infection. What are kind of the stages that a microbe, what are the things it would have to do in order to establish an infection in our body? And that will lead nicely into next week. We're just going to start a section on immunology. We're thinking about how our body is going to try to fight off those infections. So chapter 14, the tools that microbes have to infect, and then what our bodies, how it can react to that. All right, so we're going to start by talking a little bit about normal microbiota and the influence that that has. We have a lot more to learn about that, but kind of introduce that. And then think about how microbes gain access to the body, and how they usually have to attach to certain cells before they can invade further. What are some of the tools that they have in order to do that? We call those virulence factors. Briefly about host responses to that, and then how do those microbes leave our body to go off and infect other hosts. All right, and then thinking a little epidemiologically, we're going to talk about some terminology about how that's transferred to other hosts as well. So really important stuff, living in a pandemic, very applicable. All right, so thinking a little bit about our normal microbiota. Sometimes I slip and I say normal flora. That was kind of the old term that we use for kind of the microbes that are normally inhabiting our body. We kind of turned away from using that term flora because, right, flora and fauna are animals, flora are plants. Okay, the microbes in our body are not plants, so that kind of gives the wrong impression there. So more often we use the term microbiota or just microbiome. All right, so these are the microorganisms that are living in our body. Of course, we most often think of those microorganisms as being bacteria. We could have some archaea in our bodies. Of course, that domain of prokaryotes, we don't think ever causes any kind of disease, where bacteria have the potential to, but we have bacteria, archaea. There's even sometimes fungi living in our bodies that's not harming us, yeasts most likely, maybe even some protozoa, maybe even some viruses. We really, and of course, the bacteria in our bodies could have bacteriophage, viruses in our bacteria. So we really know a lot less about the viruses that may normally live in our body. We don't really have a very good inkling whether they are contributing to our overall health or whether they're just kind of hanging out there. We don't have as good of mechanisms to identify viruses that are in our body. So that's really kind of, we don't know a lot about that. But we can describe our relationship with this normal microbiota as either mutualistic or commensalistic. So in a mutualistic relationship, both members are going to benefit. Okay, so both organisms benefit. Okay, and then a commensalistic relationship is one where one member benefits and the other is not affected. Of course, if it's a parasitic relationship, then we wouldn't call that organism part of our normal microbiome. If it was harming us, then we would call it an infection. Okay. And sometimes it's difficult really to know which of these, how to describe the relationship, because sometimes, you know, we used to say, oh, the staph epidermidis that lives on our skin, that's just a commensalistic relationship. You know, we help the staph, but it doesn't really have any effect on us. But now that we know more about that relationship, we understand that it does help us because it probably outcompetes other organisms that would be likely to cause harm to us. So sometimes it's hard to say for sure which of those is going on. Okay. Okay, so how do we acquire these microbes? Sometimes they're transient, meaning that we just have them for a little while. So let's see, present in our bodies for a short time. And sometimes these microbes are considered residents. So we have them for life. We acquire them soon after we're born, maybe even during the birthing process, and we pretty much have them for life. There's some commonality between us as to what kind of microbes we have, but there's a lot of variability as well. So that's a really hot area of study right now. Like, what kind of microbes should we have as part of our microbiome to make us more healthy? Can we manipulate that to make us more healthy, or way less, or have less inflammation, or whatever? It's tremendously complicated because it's hard to study all these organisms all together at once and understand what contribution each one of them is having. So one source says it's not rocket science, it's a whole lot more complicated. So just an example, this is just part of the table in your book that talks about resident microbiota. Just some examples here, nothing to memorize, but we've got lots of bacteria, maybe some fungus in our upper respiratory tract. The digestive tract is especially our colon, right? We've got lots of microbes there, maybe even some protozoa here. So a lot of these, organisms you're not going to recognize. It's just, you may recognize some of them from lab. There are areas of our body that are considered azenic, meaning they don't normally have a microbiota. Like, they shouldn't have them present. Sometimes they do, and that causes disease. But places like our internal organs, the heart, the liver, pancreas, those places shouldn't have bacteria, central nervous system, blood, lymph. At least for now, our understanding is that they don't. But many areas of the body do. And again, this is just part of this table. There's skin, lots of other regions that do have. And we kind of have an idea of the kinds of microbes, but again, we have a lot to learn there. All right, let's get some definitions straight here before we go any further. So I've used this term already a little bit before, infection. We use that term to describe when a microbe has entered, established, and multiplied within a host. Okay, but it's not necessarily causing harm. Now, most of the time it would be, but there, our microbiota, really we could call that infection of organisms that aren't causing us any harm. And that raises an interesting question, like, okay, why does our immune system allow all these microbes to live in our body? That's a really good question. There seems to be some sort of immune tolerance there that we don't totally understand. But we do know that our microbiome is really essential to keep our immune system kind of primed. All right, we'll talk a little bit more about that later. Okay, if a microbe is causing us harm, then we call that disease. Okay, so it's causing injury to the host. So a deviation from health, whether that's infectious or not. All right. A pathogen, or sometimes we say a true pathogen, that's a microbe that's going to cause disease, even if the host is, healthy and have normal immune defenses. Typically, that's going to refer to a virus or a bacteria. And then we use the term parasite for organisms that are protozoa and larger, so protozoa and fungi. That I don't, sometimes those terms are used a little bit interchangeably, but typically, again, pathogens are the small stuff. And let's see, an opportunistic pathogen is one that's only going to cause disease to the host under a certain circumstance. So we'll talk a little bit about what can cause some of our normal microbiota to now suddenly cause disease. If that takes place, then we would call that an opportunistic pathogen. But it could just be, that the microbe gets in a different part of the body that it doesn't really belong, or there's been some trauma to the body, or some difference in, the immune status of the host. The right opportunity has arisen in order for that pathogen now to be able to cause disease. Okay, but it wouldn't under normal circumstances. All right, and these two terms, I get a little bit difficult to define the difference between them. The fact is that most of the time these terms are kind of used interchangeably, pathogenicity and virulence. If we were to use these correctly, if you talk about whether an organism is pathogenic or not, so that's the ability or the capacity of a microbe to cause disease. Okay, is it pathogenic? Typically the answer is yes or no. Can it cause disease? But in reality, sometimes we say things like, oh, it's really pathogenic, meaning it's really likely to cause disease. That's not technically correct, though. Technically, it'd be more correct to say it's pathogenic because it has high virulence. So when we use the term virulence, that's a measure of the disease-causing ability. Right. Your book goes to great pains to say, well, just because something's highly virulent doesn't mean it's a severe disease. I have looked up these terms in like every other source that I could find, and no other source defines virulence that way, because when you say something's highly virulent, that implies that the infection is going to be severe. Because if something is highly, we measure virulence based on case fatality rate. All right, so if it's highly virulent, it's more likely to kill you. So virulence implies severity of disease. If something's avirulent, it's not able to cause disease. So, or it's harmless. So I would take some issue with the way the book describes that. Take it for what you will. And then what determines whether an organism is virulent or not? It's what kind of virulence factors it has. And we're going to look at some examples of those. There's strategies that a microbe has that makes it more successful in colonizing a host or evading host defense mechanisms. Right. So getting back to that term, opportunistic pathogen. All right, so, we all have, probably thousands of different species of bacteria in our guts that aren't normally causing us any harm. They're helping us digest food, for example. There are circumstances when one of those microbes could turn on us and cause disease. So we could technically call them opportunistic pathogens. Okay, if they were introduced into an unusual site in the body. So you have maybe a really nasty infection, you got a perforated bowel, right? So your large intestine has a tear in it, and those microbes in your large intestine are going to be released into your body cavity, right? That's really bad. Your appendix ruptures, all right? That's releasing microbes in places where they aren't normally, And yes, that can be a life-threatening situation. Okay, stab wounds, right? You get bacteria from your skin, you know, deep into a wound. Like that's definitely can cause disease where, you know, if it was on your skin, it normally wouldn't. Sometimes an opportunistic pathogen can cause disease if there's a change in the immune status of the host, if our immunity gets a little bit suppressed. So what are some examples that can make our immune response be suppressed a little bit? Just something simple as getting older. We know that as we age, our immune system tends to not kick in quite as effectively. Young babies don't have quite a developed immune response, so it can kind of work both ways, too old or very young. If there's any kind of genetic defects in the host, and there is a chapter in your book that talks about immune defects. We usually don't have a chance to get to those, but there's all sorts of problems. You know, a host is not able to make a certain kind of antibody, or can't make a certain kind of cell or whatever that would normally kind of keep opportunistic pathogen in check. Sometimes we have to go on immunosuppressant drugs. Steroids, for example, to maybe treat some inflammation. Steroids are immunosuppressive. That can cause then perhaps other problems with infections we wouldn't normally get. Stress, either physical or mental. Lack of sleep, you could include that in there. I know that, you know, after I remember being a college student, after finals week, I almost always got a cold. It just kind of, you know, we know that stress causes levels of corticosteroids that can lower our immunity. And other things, you know, like malnutrition that would just tend to, you know, weaken somebody. Maybe another infection sometimes can make you a little bit weaker and less able to fight off things for a little while. And of course, this is the one we talked about, changes in the normal microbiota. If you are on a course of broad spectrum antibiotics that decreases your maybe variability of your normal microbiota, things that were normally there in really small amounts have a chance to overgrow. So like Clostridium difficile, a lot of us have that just normally in our colon and it's not a problem. But if we let that overgrow, then it can cause this really nasty diarrhea. We normally have yeast vaginally and in our mouth. And again, if we're on antibiotics, it kind of cuts out some of the other organisms, then those things can overgrow. Okay. Great. All right, what other factors can affect pathogenicity and virulence? So what factors can affect whether a microbe is successful in establishing an infection in our bodies? I like to point to, I like this graphic here because although most of you might not have got a chance to do this semester, we used to in lab let students take some media home and swab surfaces and streak on the media and see what grew up. And that was always really interesting to do. I always had a few students that were just really grossed out by this. That was my water bottle, I'm gonna throw it away. Or, you know, my countertop, look at everything that was growing on my countertop. And I always kind of referred to these three things to try to tell them to kind of, let's keep this in perspective. Okay, number one, the species of organism that was growing up there is probably not one that's going to be disease causing. All right, it's probably one that you've been around your whole life. You know, it's on the counter, you know, big deal. It's not, you know, something like MRSA, most likely. All right, also keep in mind, most infections take a certain, most microbes, you have to be infected with a certain number of them. All right, so you had very few on your countertop. We grew them up by the millions on this nice media. Like if you've got colonies on your countertop, that's probably, you should probably clean that. But, you know, we're growing them up a much higher number. oftentimes when you're only exposed to a few microbes, they're not going to make you sick. And of course, they have to come to the right, opening in our body in order to make us sick as well. We'll talk about that. And if you're generally healthy, that's, something in your favor as well. So, thinking about all of these things, and of course, if somebody, I remember what I was going to say about infectious dose. I read, well, I'll get to that story in just a second. All right, so we're going to look at all three of these things in a little bit more detail. This is not a chart to memorize. This is just some examples. This is just pointing out that some organism, they're going to have lots of virulence factors, lots of tools. And again, we'll get to some examples of those in a minute that makes them really good in establishing an infection. And other organisms are going to have very few virulence factors. So they're not likely to ever cause any kind of infection. So it's just giving some examples there. Here at the bottom of the list here, lactobacilli. Those are bacteria that you would most likely find in yogurt, right? Those are not likely to ever, under any circumstances, make us sick. Dipteroides, that's an organism that's commonly found on our skin. So you're getting it there now into some opportunists. So, we've got candida very often in our mouth, for instance, and that's only going to cause a problem, maybe if you're on some really strong antibiotics. Again, C. diff, an opportunist. Most of us would never get sick from a Pseudomonas infection, but that's a very common hospital-acquired infection, especially burn victims. They've lost their kind of first layer of defense in their skin. So those are considered opportunists. And then again, these organisms here at the top are ones that have lots of tools that make them very successful. This rabbit fever, it's pretty rare, but it's carried by the fleas of rabbits. And it's a very serious life-threatening infection, just like bubonic plague. Okay. Adults can carry whooping cough as kind of their transiently as their normal microbiota. So any adult that's caring for a baby that's not fully vaccinated yet should get a whooping cough vaccine. All right. So again, it depends what kind of organism you're exposed to, and it definitely matters how much you're exposed to. So we express that as the infectious dose. And for some infections, we have no idea what the infectious dose is because it's not ethical to, okay, with this person, we're going to expose them to 50 cells and this person 100. It's not ethical to do that. So we have to have an animal model most of the time to understand the infectious dose. And sometimes we express it as kind of the ID50. That's kind of an average of the number of organisms that, after inoculating a lot of different test animals. Okay, so anyway, the infectious dose can vary from one cell. That Q fever, that's something that's not terribly serious. It's more like a flu-like infection. Cholera, for instance, though, we think you have to be exposed to 1 billion cells. It's not too hard to do. Usually you get it from ingesting some water that's contaminated. It's pretty easy to ingest 1 billion cells. I read a case study a number of years ago about a foodborne illness. They tracked back, to a certain restaurant, and they tracked it back to figure out that is everybody that ate the garlic bread that got sick. So something in like the toppings on the garlic bread, that's kind of a weird thing. But the guy that ate 25 pieces of garlic bread was really, really ill. Okay, he had a huge infectious dose. So again, sometimes the more cells you're exposed to, the more likely you are to be sick or even to have serious illness. All right, here is I didn't post this. This is a picture of someone with an infection that is caused by extremely small infectious dose. We inhale even maybe just a few viral particles, and if you're susceptible and unvaccinated, you're likely to get this illness. Okay, this is measles. All right, something that we had problems with in this country a few years ago. That's gone down to about nothing now since we have other worries. But they say that if somebody has measles in an elevator, leaves the elevator, somebody else comes into the elevator an hour later, they can get sick with measles. So, just it's airborne. Again, really small infectious dose, and that's why we have to keep up our measles vaccination, so that because it just spreads so easily. Okay, and so the third thing that affects virulence, the strength of host defenses. This is kind of mirroring the same things that we talked about that can cause opportunistic pathogens to then turn on us and cause disease. Extreme age, malnutrition, any maybe acquired defects in immunity or something genetic, stress, if you're on an organ transplant or on some kind of chemotherapy, you're taking drugs to knock your immunity down. So again, some of the same sorts of things there. All right, so if we follow a path that a microbe might take to get somebody sick, right? So where the microbe enters the body, we call that a portal of entry. And we're talking about exogenous pathogens. So not a normal part of our normal microbiota that now becomes disease causing, but a pathogen that comes from the outside and enters one of these portals of entry. So let's go through these one by one. Skin is not a very common portal of entry. And I think that perhaps I didn't post this. We're going to look a little bit more closely at the defenses that our skin has next week. But our skin is a really good barrier. It's not a common portal of entry as long as it's intact. It's many layers. There's dead cells on the outside that are continually being lost. So when a pathogen tries to attach, it gets kind of sloughed off very easily. There's lots of defenses at the skin. So again, we're going to talk about that more. There is the potential for a hair follicle to get infected. That's probably maybe the most common thing. But as long as the skin's intact, it's not a very common portal of entry. The most common portal of entry is going to be at our mucous membranes, because the skin lining those mucous membranes is much thinner and it's alive. So, and this is also where we have a lot of our normal microbiota helps to outcompete. Probably the number one portal of entry here is going to be the respiratory tract. Okay, so you can think of a lot of infections like coronavirus that's going to target the respiratory tract. Probably the second most common portal of entry would be the gastrointestinal tract. So things that you eat, foodborne illness, you consume contaminated water. Luckily, that doesn't happen very often in this country now that we treat our water supply. But probably that's the second most common. And then urinary tract, reproductive tract, even the eyes. Okay, so sexually transmitted disease falls in there. Placenta, this is not too common. There's only a few infections that I can think of that are able to cross the placenta. German measles, it's a virus, toxoplasmosis. That's the one that, pregnant women shouldn't scoop the kitty litter because that fungus, I think it's protozoa, could be in the cat's feces. Anyway, not common. Listeria is a bacteria that can, but that's not terribly common. And the one that's probably, maybe at least a terminology that you wouldn't be familiar with, this parenteral route. So enteral, if something is being transferred by the enteric route, that enteral prefix means intestinal. So that's kind of like the normal route that you would ingest something, you would swallow it. Okay, so that prefix par means not the normal. Okay, so if you think about that as being not a normal route, okay, we include in this portal of entry anything like a syringe or a cut in the skin or a mosquito bite. Kind of like we consider that kind of not a normal portal of entry. Okay, so again, skin is good as long as it's intact, but sometimes things happen to puncture our skin. All right, so once a pathogen has gained entrance to our body at its correct portal of entry, and usually it's pretty particular, it has to come to the correct one, right? We breathe in HIV and we're not going to get AIDS. That's not the right portal of entry. It has to come to a different portal of entry. Typically, the pathogen has to then attach to its target cells. Again, that's required to successfully colonize the host. They're going to have some kind of mechanism to help them do that. And it could be some kind of physical structure like a fimbriae, plural fimbriae, or pili. Again, those structures are sometimes kind of used interchangeably. They're both made of the same protein pelin. Typically, we think about the fimbriae as being most useful for attachment, but pili sometimes is used that way as well. Parasites might have, like you think about a tapeworm, if you've ever seen a close-up of that scolex, the head that kind of anchors into our intestine in order to get nutrients from us, that's the adhesion factor for sure. And then there's typically going to be some kind of protein associated, at the ends of those fimbriae or pili, or just by itself. So there's going to be a bacterial attachment protein. And we have special names for those. We call them either ligands or adhesins. The term adhesins makes a lot more sense because you kind of, okay, yeah, it's something that adheres, but you should be familiar with that term ligand as well. Just an S there, okay. So here's just a reminder of difference between flagella and fimbriae, right? Fimbriae are much shorter, flagella are longer and used for locomotion. And there's going to be some kind of, again, protein on the end of that fimbriae that's going to attach to the right thing. Let me come back to that in a second. So here's the picture from your book that kind of, I kind of like going through and labeling a picture yourself. I think it kind of helps you direct you a little bit as to what you're actually doing. So here's a bacterial cell and here's a host cell, and it's attaching to that host cell. So on the surface of the bacteria cell, there's going to be some kind of ligand and some kind of protein that's going to recognize down here, this is the host receptor. And you might think, I've often thought, well, why do we even have these receptors? They may have some other function, all right, besides just being a mechanism for, bacteria to attach to us. That host receptor could have a lot of different compositions. It could be lipoprotein or glycolipids, some kind of sugar protein or fat. Okay. The coronavirus, of course, it has spike proteins that act kind of as ligands. And we know that it attaches to, you may have heard ACE2. These are angiotensin converting enzymes that are highly expressed in alveolar cells. So Again, even coronavirus attaches to a certain kind of receptor deep in our lungs. Capsules. So in this picture over here, E. coli, this black part here in this transmission electron microscope is a capsule secreted by E. coli. And capsules, they're sticky. They help bacteria stick to structures. We've talked about that. The E. coli is also going to have some adhesive proteins. It also has fimbriae, which you can't really see in this picture, but it has lots of strategies in order to stay anchored to the cell that it needs to attach to. And here's another organism. This is these, I think I did post this, these green rod-shaped cells here. That's Bordetella pertussis. That's the organism that causes whooping cough. And I might have forgot to say, I use a lot of examples here. Don't worry about the examples of the organisms or the disease they cause. Just trying to get through the principle that adhesion is important for cells to establish an infection. The Bordetella pertussis makes like 8 or 9 different virulence factors that help it stick to certain kinds of cells, because that organism doesn't invade any further into cells. So it stays on the outside of them. So it's really important that it stays stuck there, and it has lots of strategies in order to do that. All right, so when the microbe needs to invade further into the host. Okay, so here we're going to talk about these all. We're going to give you some examples of all these different kinds of virulence factors. Extracellular enzymes, toxins, as well as anti-phagocytic factors. So we're going to look at all of these in a little bit more detail. Some examples of virulence factors here. So let's start with extracellular enzymes. In a normal semester, you would do, if you were in 213 lab, you would do a lab where we tested for the ability of some microbes to produce extracellular enzymes. Another way to call these, we would call these exoenzymes. Right? And all cells are going to secrete or are going to have enzymes that work inside the cell, but certain organisms are able to secrete certain enzymes. Again, that helps us identify them. So the exoenzymes that we usually test for in lab are ones that help bacteria break down some macromolecule in order to use it to metabolize for energy. Like a lipase, for example, is secreted to help it break down lipids. But these extracellular enzymes are ones that can be secreted by certain organisms in order to help them invade. So some examples here, hyaluronic acid is, we could think of that as the glue that holds cells together. All right, so if the organism can produce hyaluronidase, okay, that kind of breaks down the adhesions between cells. So it allows the organism to, again, invade deeper into our tissues. Collagenase, collagen is the main structural protein that we have. And so, again, collagenase helps the organism, again, invade deeper and deeper. Okay, so a few more examples. One that will be familiar to you if you are in lab is coagulase. So not very many bacteria can secrete coagulase, but it is an enzyme that allows the organism to clot the blood around it. So it can kind of wall itself off from any non-specific immune cells that might come at it. So it can kind of hide out here, grow, multiply a whole bunch until the time is right. It has, sufficient numbers that it can break out of the clot, and it needs a second enzyme to do that, kinase. And they can, go and, it's in sufficient numbers now that it has a better chance of being successful in invasion. All right, so remember you did coagulase test if you're in lab. That's the one where you inoculated the rabbit plasma and looked for a clot. All right, so those were some examples of extracellular enzymes. Couple different kinds of toxins. Okay, what do toxins do? Well, they harm or damage host tissues. Okay, we've already talked a little bit about endotoxin. So again, hearkening back to that gram-negative cell wall structure, endotoxin is the same thing as lipid A. And if you remember, I'll come back to this slide in a minute. I don't know if you remember, come on, go to my next slide. There we go. If you remember, I don't think I posted this, but here's the gram-negative cell wall structure. Lipid A is part of that lipopolysaccharide. Okay, lipid A is the part that's really right down in there into that outer cell membrane. We talked about that again. This is the same thing as endotoxin. We used to think endotoxin came from within the cell, and obviously it's on the outside of the cell. But that is going to be released whenever the cell wall is broken up a little bit. So to some extent, when the cell divides, a little endotoxin might be released, but especially when that cell dies, the cell wall breaks down and lots of lipid A, lots of endotoxin is going to be released. Okay, and nearly all gram-negatives have this. There's different variations of it. So, some endotoxin can be, have a little different effect than others. But what kind of effect does this have? We consider it to have low toxicity. So in other words, typically this is not too much of a problem. It causes things like fever, inflammation, maybe some blood clotting. Again, typically not too much of a problem, but if you have a gram-negative infection and say maybe you take an antibiotic to get rid of that and all those gram-negatives die at once, okay, the inflammation that causes can potentially be deadly. So we have to be a little bit more careful when we're treating infections caused by gram-negative organisms. We may need to give some kind of anti-inflammatory, or oftentimes we may not treat it at all and let it run its course. And because again, the effect of all that endotoxin being released at once, it may be worse than just letting the disease run its course. Okay, all right, so the second kind of toxin is called exotoxin. Okay, and this is typically going to be a protein, often some kind of an enzyme. So hopefully you're asking the question, okay, how is an exotoxin different than an extracellular enzyme? Okay, they're both proteins, they're both secreted outside of the cell. An exotoxin is going to act systemically. In other words, a little bit of it goes a long ways, and it can act throughout, our whole body, throughout the whole host. An extracellular enzyme is going to just act right in the vicinity of the cell that secretes it. So it acts very locally. Where a toxin, again, a little of it goes a long way. Okay, it's considered to have high toxicity, meaning it can kill host cells at a really low concentration. And typically the effects are going to be very specific. Okay, so it's going to act on a certain kind of cell in the host body. And of course, this can be gram positives and gram negatives. that secrete exotoxins, where endotoxin is only gram-negative. Right, so here's some examples of exotoxins. A cytotoxin is a very general name for an exotoxin that would just kill a host cell. Okay. A neurotoxin is going to act on nerve cell function. So something like botulism toxin, that's considered to be the most potent toxic known to man. It only takes a little bit of it, extremely small amounts to kill somebody. But that toxin acts by inhibiting the acetylcholine, I believe. The nerves can't transmit. So eventually, your muscles, you can't move your muscles. Of course, the diaphragm is a muscle, so you need to move that muscle to breathe. So eventually, botulism food poisoning can be deadly. Tetanus is another example. Tetanus toxin is a very potent neurotoxin, acts a little different way. Another example is an enterotoxin. So not endotoxin, kind of sounds like it. Entero, remember, means intestinal. So this is a toxin that acts on the cells lining the gastrointestinal tract. Okay, usually produces movement of bowels go a lot quicker. So typically that's going to cause some diarrhea. All right, Staph aureus produces a pretty potent enterotoxin. It causes kind of a nasty diarrhea, food poisoning. Okay, so I kind of like this table here that compares bacterial exotoxins and endotoxins. We talked about most of these things, not heat stability. I'd say don't worry about that, but how it affects the host. Again, an endotoxin, fever, malaise, in extreme cases, shock. Does it produce a fever? That's sort of interesting. So yes, no for exotoxins. Antigenicity. So we're going to pick that word apart a little more when we get to our immunology. That just means, does it cause an immune response? Interesting enough, the endotoxin, not really. And exotoxins do. So basically what that means is we can make a vaccine that's going to be helpful against exotoxins. Tetanus vaccine, diphtheria vaccine, they are both targeting the toxins produced from those organisms that produce them. It's not possible to make, at least right now, to make a vaccine against any kind of endotoxin because it doesn't elicit a good immune response. And then just kind of some examples of diseases where that toxin production is really important. Okay, don't memorize. Okay, lastly, I want to talk about some anti-phagocytic factors. So we got to review a little bit or do a little overview of phagocytosis. We're going to pick this process apart a little bit more detail next week. But phagocytes, organisms that are able to do phagocytosis, cells that are able to do phagocytosis, white blood cells. So they are going to engulf and remove invading pathogens. Okay, so we'll talk about specific white blood cells that are really good at phagocytosis. Macrophages are an example of one that you probably heard about. Okay, so say this is a macrophage here. It's going to extend its pseudopods around the invading microbe, say it's a bacteria, and it's going to basically make kind of a membrane-bound compartment. We call that an endosome or a phagosome. So it's going to carry that microbe in a membrane, and then the lysosome will fuse with that phagosome. Remember what's in lysosomes, lots and lots of digestive enzymes. Again, we'll talk a little bit more detail about that later. Those digestive enzymes act on that microbe and most of the time, like 99% of the time, are able to kill it and then release it from the cell. The problem is that microbes have ways to withstand this. Okay, so some examples of some anti-phagocytic factors. Sometimes they are able to prevent engulfment. So one example there is just if the organism has a capsule, it makes it a lot harder for our phagocytic cells to grab a hold of that microbe and kind of, engulf it. So that's what this is showing right here. It's not, the pseudopods aren't able to grab a hold and the microbe gets away. Another strategy, some bacteria can produce leukocidin. This is a cytotoxin that basically acts to kind of punch holes in the membrane of that phagocytic cell. So, and not just evades phagocytosis, but kills the phagocytic cell. Another example is M protein. And I like to talk about this one because this is a virulence factor that Streptococcus pyogenes has. That's an organism that we use in lab. It's an organism we're going to talk about a little bit later on in the course. This stands either for membrane protein or motif protein. I've heard that it stands for both those things. But here's an example of the M protein on the outside of a strep pyogenes. Here's a cell that doesn't have the M protein. So you can see these proteins that stick out quite a bit from the surface of the cell. And basically what they do is make it a lot more difficult for phagocytes to phagocytize the cell because, and again, I'm talking about something that we're going to talk more about later, but complement protein is something that will adhere to the outside of bacteria cells and make phagocytosis more likely to happen. Those M proteins make it so complement can't adhere there. So again, more about complement later, but complement proteins can't adhere because of that M protein on the surface of strep pyogenes. So that's just an example of an anti-phagocytic factor. Another strategy is for the microbe to allow itself to be engulfed or phagocytized, but then it keeps the lysosome from fusing with the phagosome. So it can actually, hang out and live quite normally within the phagocytic cell. It's kind of like a criminal hiding out in the police station. The cell that's sent to, take care of it, then it lives happily inside the cell. And Mycobacterium tuberculosis does that. The organism that causes TB, that's what it does. It reproduces inside of macrophages quite contentedly because, again, the lysosomes don't fuse. All right. Okay, got a couple minutes left. Let's talk a little bit about thinking about, okay, so we've got these organisms that are invading, they've gotten the right portal of entry, they've attached, maybe they've invaded a little bit further. Thinking about how this reflects or how we describe these stages when we're looking at a person with disease. All right, so the stages of infectious disease. So time 0 here is when the person is infected. And then if we look at over time versus the number of microorganisms, usually the more microorganisms, usually the more severe the symptoms are going to be. All right, so typically once that microbe enters a portal of entry, we have a certain amount of time before it can, reproduce enough to cause any harm. So we call that time the incubation period. Okay, there's no signs or symptoms. And the difference between a sign or a symptom is that symptoms are felt and signs can be observed. Okay, so an example of a sign would be let's see, a fever or a rash or say vomiting. Okay, a doctor can see those things happening. A symptom might be something like a headache or some kind of pain or achiness, malaise. Okay, so in the incubation period, there's no signs or symptoms. All right, we are out of time, so I'll pick up there next time. We probably will start our immunology next time, so I'll get that next chapter posted as soon as I can. Okay, have a good day. Enjoy your weekend.